Welcome to today's deep dive. I want you to imagine your cells, right? But not as just these, I don't know, passive floating bags of water.
Yeah, or like simple chemical reactors, which is how we usually think of them.
Right, exactly. Instead, picture them as microscopic mountain climbers, like actively scaling these rugged 90-degree cliffs of scar tissue.
Literally pulling themselves up by grappling hooks.
Yeah, it's a wild visual because today we are unpacking a biological engine that acts as the secret driver behind some of the deadliest conditions we face.
Things like severe organ fibrosis and pancreatic cancer.
And more importantly, we're going to look at a newly developed drug that traps these diseases, not by poisoning them, but by just turning off the physical lights.
It really is a fundamental shift in how we view mechanobiology. I mean, for the longest time, the traditional view of medicine has been highly chemical-centric.
Yeah, we look for a chemical target.
Right. We tend to view biology as a series of chemical reactions and diffusing gradients. But this groundbreaking paper we're diving into today from Nature Cell Biology, it completely moves us away from that chemical map.
Right, because it decodes this purely physical phenomenon called durotaxis.
Exactly, durotaxis.
So let's define that core concept right away for the listener, because a lot of people tracking this space are familiar with chemotaxis, right? Which is a cell moving toward a chemical signal.
Yeah, like following a scent.
Right, but durotaxis is an entirely different beast.
It really is. So durotaxis is a highly specific type of cell migration that is driven entirely by physical stiffness gradients. Literally, it's the process of cells moving from soft tissue to stiff tissue.
Just following the tension.
Yeah. And what makes this so fascinating is that this movement is completely independent of chemical gradients.
Wow.
It's even independent of the density of the proteins the cell is crawling on, which that's a separate process called haptotaxis.
Okay, so it's not following chemicals, it's not following protein density.
No, durotaxis is a purely mechanical reaction to just the firmness of the ground beneath the cell.
Okay, so before we can understand how these cells climb, we first have to understand the terrain they're moving on.
The physical landscape.
Right. So the researchers wanted to measure the actual mechanical landscape of a living organ. But, I mean, how do you even go about measuring the physical topography, like the literal squishiness of a microscopic lung?
It's a great question. They use this brilliant piece of analytical tech called atomic force microscopy, or AFM for short.
Okay, AFM, oh.
Yeah. And instead of using optical lenses to look at a tissue, AFM uses a microscopic cantilever with an impossibly sharp tip.
Like a needle.
Conceptually, yeah. Think of it like a microscopic diving board with a tiny needle at the end. The machine physically presses that tip into the tissue.
Oh, wow. Literally poking it.
Exactly, poking
Yeah.
it. And then a laser is aimed at the back of the cantilever, and it measures exactly how much that microscopic diving board bends when it pushes into the organ.
So the stiffer the tissue, the more the board bends.
Yeah. You got it. And by calculating that deflection, it gives us the Young's modulus, which is just a precise measure of stiffness calculated in units called pascals.
Pascals, right. And just to give everyone listening a clear visual, they didn't just poke the tissue blindly. They combined this physical probing with a specialized dye called picocereus red, right?
Yes, that specific stain binds to collagen.
Which is the main structural protein in scar tissue.
Exactly. So they could literally map the visual location of the scar and then feel the exact physical stiffness of that scar at the exact same time.
That's incredible. So they mapped the lungs, the skin, and the kidneys of mice. What did a normal, healthy organ look like to this tiny needle?
Well, in healthy, uninjured tissue, the mechanical landscape is remarkably uniform. It's essentially just a series of gentle, rolling hills.
Like a flat meadow.
Yeah. If we look at their baseline data, the slopes, meaning the rate at which the stiffness changes over a microscopic distance, they're incredibly gentle. Healthy lungs had an average slope of around 47 pascals per micrometer.
Okay, 47.
The skin was about 69, and the kidneys were 29.
So mechanically speaking, the cells are just strolling through a flat, grassy park.
Right.
But then the researchers purposefully triggered fibrosis, right? They used a chemical called bleomycin to severely damage the lungs and skin, and a surgical procedure called ureter ligation for the kidneys.
To induce that severe scarring, yeah.
And when they remapped those damaged organs, the topography just violently shifted.
The transformation was dramatic. The fibrotic tissue displayed extreme heterogeneity. We're talking about Microscopic focal peaks of stiffness reaching all the way up to 40 kilopascals.
Wait, 40 kilopascals, so that's 40,000 pascals.
Exactly, 40,000, and then deep valleys dropping all the way down to like 0.5 kilopascals or 500 pascals.
The gradients are just what blow my mind here, the cliffs. Because the fibrotic lung went from that gentle slope of 47 pascals per micrometer to a staggering 500 pascals per micrometer.
Massive jump.
Yeah, the skin shot up to 433 and the kidneys hit a 115. To put that in perspective for a cell, that's the difference between walking on a flat sidewalk and suddenly confronting a jagged, vertical cliff face.
It's an intensely hostile physical environment.
Mm-hmm.
Biologically speaking, these gradients in the diseased tissues are up to 1,500 times steeper than the stiffness gradients you'd naturally see during normal embryonic development.
Just unbelievably steep. Okay, so to just do a quick recap for you listening, AFM revealed that our fibrotic organs have these massive steep stiffness gradients compared to healthy organs.
Right.
They form physical peaks and valleys reaching up to 40 kilopascals. So the question for you is, have you ever thought of your organs as having a physical topography at the cellular level? And what if diseases are exploiting that exact map?
Because they absolutely are.
Right. Which brings us to the next point. If you knew your tissues had this extreme terrain, why would your cells actively choose to climb the steepest, most difficult parts of it?
Well, to answer that, we have to look at how different types of cells react to that specific terrain. So the researchers had to transition from the living organs back into the laboratory.
Right, because you can't easily watch a single cell climb a microcliff inside a breathing mouse.
No, you definitely cannot. So they recreated these microscopic mountains in a petri dish.
How did they even do that?
Two incredible engineering feats. First, they used a technique known as photolithography.
Okay.
They took polyacrylamide hydrogels, PA hydrogels, and used precise UV light exposure through a microscopic mask to create alternating stripes of varying stiffness.
So they literally printed the stiffness.
Exactly. They printed one soft stripe at 4 kilopascals, sitting directly next to a stiff stripe at 40 kilopascals, and they just repeated that pattern over a span of 40 micrometers.
That's like printing a microscopic pedestrian crosswalk, but instead of white and black paint, it's soft and stiff gel.
That's a perfect analogy. And their second feat used microfluidic gradient generators. This let them create a continuous 1 millimeter long physical slope.
To mimic the lung.
Right? It perfectly mimicked that steep 500 pascals per micrometer gradient they saw in the fibrotic lungs. And the chemical environment was kept perfectly stable. Only the physical stiffness changed.
Okay, so when they placed different types of cells onto these engineered crosswalks and ramps, the sorting behavior was wild.
It was very distinct.
Yeah, they calculated something called a durotactic index, right? Which is basically a ratio of how many cells actively chose to migrate to the stiff side versus the soft side.
Correct. And mesenchymal stem cells, or MSCs, and fibroblasts, which are the cells that build tissue and generate scars, they showed massive durotaxis.
Massive. Like their index was greater than 6?
Right. Meaning they overwhelmingly flocked straight to the stiffest stripes. It's a profound migration.
But then endothelial cells, the ones lining blood vessels, and epithelial cells, they only showed moderate durotaxis.
Yeah, and they mostly only managed it when they were moving collectively in clusters, not so much as single cells.
But the part that fascinated me the most was the immune cells.
Oh, the macrophages.
Yeah. The M1 and M2 macrophages, the T cells, their durotactic index was essentially just a 1.
Right, they completely ignored the stiffness.
A 40 kilopascal cliff meant absolutely nothing to them. They just wandered randomly across the soft and stiff zones. But wait, why? Why do fibroblasts compulsively climb these cliffs while immune cells just couldn't care less?
Well, think about the evolutionary logic of an immune cell's job. It's a first responder, right?
Okay.
A macrophage has to patrol everywhere. It has to move fluidly through soft fat, dense muscle, and stiff scar tissue alike. If an immune cell got biologically trapped by a physical stiffness gradient, it would be completely useless at fighting an infection located in, say, an adjacent soft tissue.
Oh, it would just get stuck on the scar.
Exactly. Fibroblasts, however, are the body's structural builders. They are programmed to seek out mechanical tension because, historically, tension means there is a structural failure or a wound that urgently needs reinforcement.
That makes perfect sense. They feel the tension and go to fix it.
Right. And to prove this actually happens inside a living animal, the researchers use a specialized genetic tool called a COL-GFP reporter mouse.
Okay, explain the reporter mouse.
So in these genetically engineered mice, Anytime a cell turns on the genes required to produce collagen, specifically high Col1A1 or ACT8-2, meaning it's actively transforming from a normal cell into a scar-forming myofibroblast.
Yeah.
It brilliantly glows green under a fluorescent microscope.
That's so cool. So they took these living fibrotic lungs and used FACS fluorescence-activated cell sorting to perfectly separate the glowing green scar cells from the normal ones.
And what the physical mapping showed perfectly mirrored the lab results.
The soft valleys contained the normal, unactivated fibroblasts.
Right, GFP low.
But the stiff peaks were entirely packed with the activated, glowing green myofibroblasts.
Exactly.
Okay, but here is the massive plot twist in the data. For a long time, the medical assumption was that scar tissue grows because the bad scar cells sitting on the stiff site are just rapidly multiplying.
Right, that was the standard belief.
But the researchers used a molecular marker called BRDU, right, which highlights cells that are actively copying their DNA to divide.
And the data directly contradicted that old assumption. They found that less than 1% of the activated glowing green myofibroblasts up on those stiff peaks were proliferating.
Less than 1%. They were barely multiplying at all.
Exactly. But down in the soft valleys, over 6% of the normal fibroblasts were actively dividing.
So let me make sure I have this sequence exactly right, because this feels vital. The cells are not multiplying at the top of the stiff scar. They multiply down in the soft, healthy tissue.
Right.
Then, driven by that physical urge durotaxis, they hike up the steep stiffness gradient. And it is only after they reach the absolute peak, after they physically feel that extreme structural tension, that they mutate and activate into scar-forming tissue.
It is a beautifully clear mechanical sequence. The physical tension itself is the necessary trigger for the cellular transformation.
Wow. Okay, so a quick mini recap. Fibroblasts love to migrate towards stiffness. Immune cells ignore it. And fibroblasts proliferate in soft tissue, climb the mountain, and then transform into scar tissue.
Yes.
So to the listener, think about this. If the cells only turn bad after they climb the mountain, what if we just cut off their climbing gear?
Which is the million-dollar question.
Right. But that brings up a glaring mechanical question for me. A cell doesn't have hands. It doesn't have a central nervous system. How does a microscopic structure physically feel that the ground beneath it is getting stiffer?
Well, it relies on a highly specialized protein complex. It acts as a dedicated mechanosensory module.
Okay.
The study drills down into two specific proteins to explain this. FAK, which stands for focal adhesion kinase, and paxillin.
FAK and paxillin. Let's visualize the mechanics of this, rather than just saying they bind together. The cell has these structures called focal adhesions on its outer membrane, right? I picture them like microscopic grappling hooks that the cell throws down to physically attach itself to the tissue.
That's a really highly accurate way to view it. FAK is a catalytic enzyme that sits precisely at those anchor points.
Okay.
And the researchers discovered that FAK uses a specific structural region, known as the FAT domain, to physically bind to two targeted regions on the paxillin protein, specifically the LD4 and LD2 domains.
So if FAK is the grappling hook actively gripping the rock face, paxillin is the tension cable snapping taut.
Yes. When the ground is stiff, the cell tries to pull itself forward, the stiff ground heavily resists, and that resulting physical tension stretches that protein complex.
And it acts as a mechanical transducer.
Exactly. When that FAK-paxillin connection pulls tight enough, it triggers a cascade that activates a downstream protein called YAP.
YAP.
Right. YAP is essentially the genetic steering wheel. It travels deep into the cell's nucleus and physically flips the genetic switches required to drive the cell further up the gradient.
The physical tension on the outside is directly translated into a genetic command on the inside. That is incredible.
It really is.
But I gotta push back here for a second. Biology is incredibly interconnected, right? If we go in and sever that tension cable, if we medically stop FAK from binding to paxillin, won't the cell just completely shut down?
You think so?
Because it still relies on FAK for basic survival and to move around to find nutrients using chemical signals, right?
And that is the most elegant revelation of this entire paper. The researchers tested this by using cells with a highly targeted genetic mutation to the paxillin protein.
Okay, what kind of mutation?
They use these specific paxillin mutants.
Uh-huh.
One is called PXNY31118F, which is a mouthful, but basically it's a non-phosphorylatable version.
PXNY31118.
Got it.
This specific mutation prevented FAK from locking into it. So it effectively severed that specific tension cable. And when they placed these mutated cells onto the engineered hydrogels, their durotaxis was completely abolished.
They were physically incapable of climbing the stiffness gradient.
Exactly.
But what about their chemical sense? That's the brilliant part. When they tested those exact same mutated cells for chemotaxis moving toward a chemical signal, they moved perfectly normally.
No way.
Yeah, and when they tested them for haplotaxis moving toward a denser protein coating, they moved perfectly normally.
That is just, it's a highly segregated biological circuit.
Extremely segregated.
Okay, recap time. Cells feel stiffness through the FAK-paxillin protein interaction, and this pathway is entirely separate from how cells sense chemical signals.
Correct.
So, question for you listening. Imagine having a completely separate nervous system just to feel the gravity of the room you're in. How would you use that information? Because our cells are using it to build diseases.
Which leads us directly from fibrosis into oncology.
Right, because so far we've seen how this mechanism builds a localized scar in a misguided attempt to heal a severe wound. But what happens when a rogue mutated cell realizes it can use that exact same scar tissue, not to heal, but to build a physical highway to escape the organ entirely?
And that brings us to pancreatic ductal adenocarcinoma, or PDA.
PDC, which is pancreatic cancer.
Yes, and it's notorious for being incredibly aggressive and highly metastatic. A defining hallmark of PDA is a biological phenomenon called desmoplasia.
Desmoplasia, meaning it creates a dense, highly fibrotic tumor microenvironment.
Yes, the TME. The tumor effectively forces the body to build a massive fortress of scar tissue around itself.
So to study the mechanics of this, the researchers used a highly specialized mouse model, right? They took SCID mice.
Right, SCID mice. They're engineered without an immune system, so they won't reject human tissue.
And they orthotopically injected them, meaning they injected the cancer directly into the actual anatomical pancreas, not just under the skin.
Crucially, they co-injected a GFP luciferase human pancreatic cancer cell line alongside M-cherry cancer-associated fibroblasts, or CAFs.
CAFs, and those CAFs are the hijacked cellular architects, right?
Exactly. Their sole biological job is to constantly build that stiff fibrotic matrix around the growing tumor.
And when they analyzed these growing tumors, the AFM needle found those exact same steep, extreme stiffness gradients right at the tumor's invasive front.
Right at the border between the malignant tumor and the healthy host tissue.
So they recreated this specific microenvironment in the lab using hydrogels, like 1 kilopascal soft to 10 kilopascal stiff. And they literally observed clusters of cancer cells surfing the stiff stripes out of the main tumor body.
Cancer cells were actively utilizing durotaxis to migrate along the stiffest mechanical paths created by those corrupted fibroblasts.
But okay, here is where I hit a bizarre contradiction in the source text.
Okay.
The cancer cells are clearly using durotaxis to move from the relatively soft core of the tumor to the incredibly stiff outer edge. They are biologically addicted to climbing stiffness.
Right.
But metastasis requires them to eventually leave the tumor entirely, enter the fluid bloodstream, and invade entirely different organs like the liver or the GI tract, which are comparatively soft.
Very soft, yes.
So why would a cancer cell, strictly programmed to continually climb towards stiffness, suddenly decide to jump off the cliff into soft tissue?
It's a phenomenal biological paradox, and it highlights a critical mechanism known as a mechanosensory switch, or desensitization.
A switch?
Yeah. Durotaxis acts strictly as the initial launch pad. The steep stiffness gradients perfectly guide the cancer cells to the absolute outer boundary of the tumor mass, breaking them free from the main body.
Okay.
But once they reach the absolute peak of that stiffness of the tumor boundary, the extreme physical tension forces a massive cytoskeletal reprogramming within the cancer cell.
Oh, so the sheer mechanical stress of the peak forces them to adapt.
Exactly. The extreme tension triggers a desensitization. They intentionally switch off their durotactic dependency.
Wow.
And this sudden cytoskeletal shift is what allows them to survive the intense fluid shear forces of the bloodstream and successfully colonize the softer distal organs.
So the stiff scar isn't a defensive wall keeping the cancer contained. It's a high-speed rail network explicitly designed to launch the cancer cells out into the rest of the body.
That's a terrifying but accurate way to look at it.
Quick recap for the listener. Pancreatic cancer uses stiff fibrotic tissue as a highway to escape the primary tumor. They use durotaxis for early dissemination, and then they switch behaviors.
Yes.
So the question to you listening, could the key to stopping cancer metastasis be not attacking the cancer cell itself, but simply softening the highway it uses to escape?
Which brings us directly to the therapeutic interventions they developed. We know the exact engine driving this, the FAK paxillin tension cable.
Right.
And we know it drives the lethal progression of both organ fibrosis and metastatic pancreatic cancer.
So the ultimate challenge was figuring out how to break this specific engine in a living creature without causing lethal systemic side effects. Because we noted earlier that FAK is essential for other cellular functions.
Exactly. A global genetic knockout, meaning entirely deleting the FAK or paxillin genes from an animal, is embryonically lethal.
They just won't develop.
Right. An organism literally cannot develop without those baseline catalytic functions. So the researchers had to be incredibly surgical. They engineered a knock-in mouse model.
A knock-in mouse?
Yeah. They mutated just a few highly specific amino acids in the genetic code to create the FAK FATL994E mouse.
FAK FATL994E?
This incredibly precise mutation solely disrupts the exact binding site where FAK physically locks into paxillin for stiffness sensing. Every other function of the protein remains perfectly intact.
And the physical result of that precision? These mutant mice are born perfectly healthy.
Really normal.
They develop normally, they live completely unimpeded lives, which conclusively proves that durotaxis is not strictly required for daily baseline biological homeostasis.
Right. But when the researchers exposed these specific mice to the lung-scarring chemicals we discussed earlier.
The lutea.
Exactly. The resulting fibrosis severity absolutely plummeted. The cells simply lack the physical capability to feel the structural injury, so they never migrated to build the massive scars.
It's a stunning proof of concept. But obviously we can't easily go in and rewrite the foundational DNA of human patients currently suffering from these diseases.
Right. Genetics is tricky in living patients. So the researchers utilized those findings to test a highly targeted pharmacological intervention, a small molecule inhibitor named JP153.
JP153? How does that drug work physically?
It acts as a direct chemical mimic of that exact genetic mutation. When administered, JP153 wedges itself deep into the structural pocket of the FAK protein, physically blocking it from connecting to paxillin.
But it doesn't stop FAK's other jobs.
Crucially, no. Just like the genetic mutation, it does absolutely nothing to stop FAK's normal, necessary catalytic enzyme activity. It simply cuts the physical communication line for stiffness sensing.
Okay, so they took this novel drug, JP153, and tested it on the highly lethal PDAT pancreatic cancer mouse model. What were the actual results?
The results were profound, and they really challenged traditional oncology metrics. The primary tumor size in the pancreas stayed exactly the same.
Wait, it didn't shrink?
The drug did not shrink or kill the main cancer mass at all. However, the metastatic burden, the aggressive spread of the cancer into the liver and the GI tract, was drastically significantly reduced.
Wow. Let's really put that in perspective. For decades, the entire field of oncology has heavily focused on developing highly toxic chemical compounds designed to aggressively poison and shrink primary tumors, right? Which causes devastating systemic sickness in patients.
Yes, chemotherapy is brutal.
But this new drug, JP153, it doesn't actually kill the cell. It acts as a molecular blindfold. It physically traps the cancer in the dark, cutting off its spatial awareness so it simply cannot find the stiff escape route.
It's acting like a molecular fence.
So final recap. Genetic mutations targeting only the FAK paxillin bond prevent fibrosis without harming normal development. And the drug, JP153, successfully traps pancreatic cancer cells, reducing metastasis without affecting the main tumor mass. So for everyone listening, ask yourself, we've spent decades trying to poison cancer cells into submission. Is it time we just lock them in the dark instead?
When you synthesize all the data we've discussed today, the major paradigm-shifting takeaway is that durotaxis is no longer just a quirky, interesting biophysical observation isolated to a petri dish.
Right.
It is a fundamental in vivo driver of massive disease progression. And by successfully identifying this precise therapeutic window, proving that pathological durotaxis can be safely and pharmacologically turned off without harming the organism, this study genuinely opens up an entirely new field of medicine. We are stepping firmly into the era of mechanotherapeutics.
Mechanotherapeutics, the idea of healing the body by altering physical forces rather than just chemical balances.
Exactly.
I want to leave you, the listener, with a final highly provocative thought drawn directly from the concluding remarks of this research paper. The scientists hint that this specific FAK paxillin tension cable is likely just the very tip of the iceberg.
Oh, absolutely.
There is a vast, mostly unexplored mechanosensory landscape operating inside us at all times. They point toward other potential potential biophysical sensors, things like SRC kinases,
Yeah.
piezo ion channels, which actually won a Nobel Prize recently for their role in touch sensation, and even mitochondrial fission regulators that respond to cellular stretching.
It's a whole new world of biology.
It is. Our cells are not just squishy, passive chemical bags. They are highly active, dynamic structural architects. They are constantly feeling, pulling, and reacting to the microscopic physical tension of our bodies.
They're feeling the gravity of their environment.
Right. So if extreme physical stiffness is what actually causes these complex chemical diseases to spiral out of control in the first place, does the future of treating chronic disease lie in physical structural cures rather than just chemical ones? What other massive biological mysteries are hiding in plain sight, just waiting for us to finally learn how to feel them? Keep diving deep.